Um, so I really appreciate the opportunity to tell you about the work that we're doing in the lab, which really began, over 30 years ago, when I returned back from 32 years ago, when I came back from postdoc and started at the medical school of Hebrew University in ICRF. And I actually got my first grant from the ICRF, a Research Career Development Award. And it was to help me understand how RNA binding proteins play a role in frog development. We were looking at that point at frog oocytes, and we were interested in understanding how RNAs get localized within the oocyte even before fertilization. And ICRF understood that there was a lot of importance in understanding how to spatially organize a cell, and in particular an oocyte, and that by understanding how to provide polarity within a cell, you could begin to understand perhaps how you lose polarity in situations like cancer. And so we actually found a protein that was important in localizing RNA and helping to set up that polarity within the cell, or interpret the polarity within the cell. And it turned out, though, that frog protein was very highly conserved across evolution, and that it's also expressed, or a very close cousin of it is expressed in humans, and is what we call an oncogene. A oncofetal protein, which means that the protein is expressed during embryonic development. It gets turned off after birth, and then gets turned on again in many different kinds of cancers. So we began to try and understand what this protein was doing, both in a developmental context in the during the development of the embryo, and then also in cancer. And it turned out that this protein plays an important role in when cells become transformed, become cancerous. It plays an important role in stabilizing many RNAs and regulating their translation, and essentially promotes an oncogenic program. So when the cell becomes cancerous, it turns on this protein, and then this protein helps develop that cancer program within the cell by binding many different kinds of RNAs that promote that program. And we did some mouse studies to try and look at what that protein was doing and what would happen if we inhibited the protein. And we saw that in mice, when we inhibited the protein, we could inhibit metastasis, and that the protein was also important in tumor progression. So when we saw that in mice, in lung carcinomas, we were doing that work in lung carcinomas, we said this would be a great target for a directed inhibitor, what we call, a targeted therapy. And so we did a high throughput screen for molecules that would inhibit the binding of this protein to an RNA which encodes an oncogene, a gene which helps promote cancer. And we were able to identify, out of 27,000 molecules that we screened, we found one that inhibited the protein, inhibited the binding. And what we're now doing is optimizing this molecule, and under the auspices of the ICRF grant that I just received in September, we started in September, we're hoping to test this molecule in mouse models and hopefully develop it to the point where we can take it into clinical trials. And the molecule, we're particularly excited about this idea because the molecule inhibits this protein, which binds a number of different RNAs and through different pathways promotes cancer. So we think that it'll have a very strong effect, this molecule, in terms of inhibiting cancer through a number of different avenues. It'll help prevent proliferation of the cells. It'll help apoptosis. It'll cause cell death. It'll also prevent chemo resistance to other, to chemotherapies. And prevent metastasis. So we think that because we're hitting a protein which is like a hub and helps promote a whole cancer program, we think by hitting that protein, we can have a profound effect on lung carcinomas and actually other kinds of carcinomas as well. We've seen it in a number of different kinds of cancers. So I guess that's my story right now. And it's been, very wonderful working with the ICRF over the years. We've been lucky enough to get a number of different grants from the ICRF. And this is a story which has taken a long time to develop, but we're excited about, pushing it forward now.